Hi there guys, and welcome to another lesson of A-level biology with myself, Mr Phillips. Today we're going to continue going down the year 12 content. So today we're going to be looking at building proteins. So this is following on from the previous lesson I did, which was about amino acids and how condensation reactions allow one amino acid and one amino acid to form a dipeptide. So let's get into it, the 4 levels of protein structure. So basically, if we're looking at making proteins, hopefully you already know from the last video, which we summarised, many amino acids join together via condensation reactions where water is removed to make a polypeptide chain. The bonds between them are dipeptide bonds. And a protein might just be one individual polypeptide, or it could be several joined together. And we'll talk about hemoglobin later on. So what we start off with then is the primary structure of a protein. Now, the primary structure basically means the sequence of amino acids. So if you're asked in an exam, describe the primary structure, it's a sequence of amino acids that are joined together via peptide bonds. So just to remind you, if you haven't watched the previous video, which you'd need to fully understand it, we'd basically have one amino acid here, which there's an amino group or amine group at the end. So remember, that could be called amino, or it could be an amine group. Okay, we've got another amino acid here, and basically every NCC in the middle backbone is another amino acid. And basically these 4 amino acids in this chain stick together, and in the polypeptide, therefore, you always have an amino group at one end, and you will also always have the acid or the carboxylic acid group at the other end. So basically that is that, and the structure of the protein is determined by this unique sequence of amino acids, and this is called the primary structure. And what gives it its importance is basically the R group of each amino acid. So if we look at each amino acid, if you remember that each amino acid has got 2 bits around its alpha carbon atom. So each amino acid will have a hydrogen, which you can see at the bottom of all these amino acids here. So there's one hydrogen, another hydrogen, another hydrogen, but the other side is the R group. So this amino acid here is a glycine because it's only got one hydrogen, it's got a hydrogen as an R group. But if you look at this one, it's got a much bigger R group there, and again this one at the end here, much bigger R group. And it's the R group that gives the characteristics of the protein. So for example, if you've got a lot of amino acids that are hydrophilic, it might pull out more. If you've got amino acids that are hydrophobic, it will push you away from water more. So we'll talk about that as we go through. We'll talk about it now. So let's have a look at the effect of that. So basically, the primary structure is a sequence of amino acids. Okay, it is coded for by the order of DNA bases. So you might not have done DNA before, but I'm pretty sure you should know by now that DNA has 4 bases which code for all our proteins, basically for all our characteristics of life. The bases are adenine, thymine, cytosine, and guanine, A, T, G, C. I haven't drawn that very well. C and G always go together, A and T always go together. So if you have a look at this amino acid sequence here, this DNA sequence here, you can see if we go over to this end, when you've got these ones here, so if we just look at one part of the DNA, you'd have GTC. And that codes for the amino acid glutamic acid, okay, which is a hydrophilic amino acid, I believe, meaning its R group pulls outwards towards liquid. If you therefore get a mutation, and that changes, so if you notice here the GTC has now become GAC, that then has altered the amino acid that triplet code codes for, and it becomes valine. Now, valine is hydrophobic. So what happens is when there's a lack of oxygen, the hemoglobin will basically collapse in on itself, and therefore it will lose the shape and become this sickle cell shape. Hence the name sickle cell anemia. So what this shows us is the importance of the amino acid sequence, that primary sequence, because a mutation, which is a random change in the DNA, can alter that amino acid sequence, that primary sequence, and that can have a large impact on the protein. So what would have been a normal red blood cell, erythrocyte, has now become a sickle cell red blood cell, and that can obviously cause a lot of health issues to the sufferer. So basically there is a simple code. There is only 20 amino acids. But if you think about it, that means even in this just 4 amino acids in a chain, you've got 1 in 20 that one can be, any 1 in 20 that one can be, any 1 in 20 that one can be, and any 1 in 20 that one can be. And that gives huge variety, especially when some amino acids are over, some polypeptides might be 1,000 amino acids in length. With just 4 amino acids, you've got basically these combinations. You've got 20 varieties possible there, with 20 varieties there, with 20 varieties there, with 20 varieties there, which gives you over 160,000 different combinations. If you don't believe me, have a go and then let me know. Small proteins will even contain more than 100 amino acids. Before you can have over 1,000 amino acids in larger proteins. So then we go on to something called the secondary structure. So once the primary structure, which is the sequence of amino acids, has been sorted, what happens is the amino acids basically arrange themselves into one of 2 ways, called the secondary structure. And it's determined by what happens in the primary structure, and it's caused by hydrogen bonding and a lot of interactions between the R groups. So it will depend on the sequence of amino acids. So what we can have first of all is we can have something called an alpha helix, which is this sequence here. Okay, and basically alpha helix is where there is a helical shape for the amino acids, and they make this helical shape structure rather than just being in a straight line. And you're expected to know that basically every 10 turns has 36 amino acids in it. So each turn is about 3.6 amino acids, and basically hydrogen bonding forms this. If we go on to the second example, this one is called a beta-pleated sheet. So if the amino acid chain doesn't form a spiral, it forms a beta-pleated sheet, okay, which again is due to hydrogen bonding, depending on what the R groups are, depending on what amino acids are in there, okay, but basically instead of going spiral, it will do more of a rigid up and down sort of structure. So if we look at that from a different point of view, just sort of give you the idea, we've got here the primary structure of a protein, so the order of amino acids, and what we're saying is depending on what the amino acids will be, will depend on whether it becomes an beta-pleated sheet, which you're pointing at now, which is the up and down structure, or an alpha helix. And that is due to hydrogen bonding between the amino acid sequence. So then we go on to the tertiary structure. So tertiary structure is one that you'll often get questions about in the exams. So tertiary structure is once we've got these alpha helixes, or once we've got these beta-pleated sheets, what does the overall structure lead to? So if you look at this example here, above my pen right now, that makes it look like it's in a straight line. But in real life, it wouldn't be in a straight line. Okay, so in real life, it would fold in on itself. So even if you had an alpha helix, just alpha helix on that, okay, it wouldn't be in a straight line on that. What we're saying is it would go in a sort of whatever structure is needed for the enzyme. And if you had, if it was an enzyme, for example, if you had a beta-pleated sheet like that, it wouldn't be in a straight line. What would happen is you would get your structure to be much more 3D shape. So the tertiary structure is the 3D shape of the 3D folding or the 3-dimensional shape of an individual polypeptide chain, which gives the protein its overall shape. So here we have just a different diagram of it. It just shows the complex folding of it and how it can happen. So basically there are 4 bonds, or 4 groups of bonds, I suppose, that decide the protein structure of, the tertiary structure of a protein. So if we have a look at this, basically there's 4 different bonds that we're going to be sort of looking at. So first of all, we've got ionic bonding. So if you remember from GCSE, we only need GCSE level knowledge here because we're not doing A-level chemistry, it's A-level biology. You may well obviously do this if you do A-level chemistry as well. Ionic bonding is between atoms, and it leaves you basically with a positive charge and a negative charge. So when you've got positive and negative, those two will attract to each other. We've got something called a disulfide bond, which can only occur with certain amino acids that have 2 sulfurs in their, a sulfur in their R group, allowing the amino acids to join. And then you have hydrogen bonding, which we'll talk about in another video when we do about water. So the slightly positive OH and the slightly delta positive OH, I should say, and the delta negative oxygen attract to each other. And then we have hydrophobic and hydrophilic interactions, which again determine the shape of the overall protein. Now, this leads to basically two types of protein types, or two types of, I suppose, polypeptides in the tertiary structure. You can have globular structures, which are spherical, or you can have fibrous, or something we call fibrous structures, which are long and thin. The globular ones would make up things like that have more biochemical functions, such as hormones that have to be a specific shape to fit into a receptor, or enzymes which have the very specific active site shape. And basically they have the following properties always. They would be soluble, they have a large variety of amino acids, and they have a precise shape so they can be denatured. That as opposed to the fibrous proteins, which have structural functions, so often like your hair, so collagen, things like that, and they're like much more strong and therefore long lasting, and don't rely so much on a variety of amino acids. So examples include keratin, which makes your hair, collagen, which makes your tendons, cartilage, which obviously makes stuff like your ears and your nose. Okay, and because they're structural, they make sense that they're insoluble, so they won't dissolve. They've often got repeated amino acids, often the amino acid glycines involved because it's got the smallest R group. I think collagen means 30% of it is a glycine, and as a result, they will bond. You can see these long, closely linked polypeptide chains. That means they are resistant to temperature and pH. So here's a question. So I have a quick question. We haven't done this in many of our videos, but just want to do it in this one because I think it's such an important area, this topic. It comes up all the time. People never do that well at it. So it says, each amino acid has a different R group. Describe how these R groups can interact to determine the tertiary structure of the protein. So effectively, if we have a look at a mark scheme to this, you can see that basically this just wants you to list down the different bonding structures, and because they've said about the R groups, I think some people might have got a bit confused with it. So the idea is that some R groups might attract or repel each other, but the key thing is the different bonds that you can have. So disulfide bonds or disulfide bridges would give you a mark. Okay, now we mentioned that only occurs in some amino acids, and the amino acid that has sulfur in the R group is cysteine. So that's always going to be worth an extra mark if you can remember that. Then obviously there's hydrogen bonding. Okay, and you don't really need to say much about that. There's ionic bonding between positive and negatively charged R groups, and you've got hydrophilic R groups that tend to be on the outside of the molecule, whereas hydrophilic R groups tend to pull towards the inside of the molecule. So basically what we're saying, this is hemoglobin, for example. Hydrophilic R groups will sort of pull out to maintain the structure of the hemoglobin molecule, whereas hydrophobic R groups tend to pull in to the center, and obviously that balance of R groups gives the overall structure of the protein. So far we've come up with the different information of primary structures. At the top you've got here primary structure, which again, just keep emphasizing, is a sequence of amino acids. At one end of the polypeptide you've got the carboxylic acid group, at the other end you've got the amine or amino group. And that might have a mixture of alpha helix and beta pleated sheets in it. We've then got the alpha helix and beta pleated sheet, which again, remember, alpha helix is just this sort of structure, whereas a beta pleated sheet is more of a ridge structure, which just depends on the R groups and the hydrogen bond that forms. Those then go into more complicated 3D structure, which gives us our tertiary structure. But at the minute we've only discussed, well, what if it's just one polypeptide? But actually a lot of proteins consist of more than one polypeptide. So how they interact becomes the quaternary structure, the fourth level of structure. We'll get there eventually. Right, there we go. So if we have a look at the last level, then, is the quaternary structure. So basically the structure of proteins often have more than one polypeptide. The chains are often held together by the same bonds in the tertiary structure. So that could be a question that comes up that people don't always realize, is what tends to hold together the quaternary structure of polypeptides is the same bonding as the tertiary structure. So here we've got hemoglobin. Now hemoglobin has a quaternary structure because it has four polypeptide chains, and if you're asked to describe the quaternary structure, it has two beta chains. That isn't beta pleated sheets, it's a separate meaning, beta chains, and two alpha chains, okay, which means it's got four polypeptides all bonded into one. But there's an extra level of quaternary structure with hemoglobin, and that is it has something, you can't consider the word in here, called a prosthetic group. So a bit like if I had an artificial hip or something, you might call it prosthetic hip, because it's a inorganic part of an organic molecule. So in hemoglobin, you'll be probably aware is that there is an Fe ion, okay, so ion, and the Fe ion is the thing that accepts oxygen in hemoglobin and gives it its affinity for oxygen. So that is called the heme group, and that makes up the quaternary structure. The quaternary structure is when there's more than one polypeptide and any extra inorganic bits. So how could all this come together in a question? So this video is running a bit long, but I think it's really important to do this. So if you've got a question like this which says hemoglobin is a globular protein. Now, depending on what example you do, depends on how much you need to know specifically about hemoglobin, but it's a really common answer question either way, because what it says here is describe the structure of hemoglobin, includes secondary, tertiary, and it does say quaternary structure, you can't see it, of the molecule, and it's a 7-mark question. So how would I answer this? Well, even though it says secondary, tertiary and quaternary, I would automatically mention primary structure. How could you answer this question without it? Okay, so it's only got to get 7 marks. So first thing I'll be writing is that the primary structure is a sequence of amino acids, and always mention that they are joined together by peptide bond. You may get an extra mark for saying by condensation reactions. You might not, because it's not that applicable to the question. Then we need to mention, right, well, this is a what is the secondary structure? So again, describe the fact that secondary structure is alpha helix or beta pleated sheets. Now in hemoglobin, which you wouldn't be expected to know for AQA, maybe for OCR, is that it's more alpha helix and smaller regions of beta pleated sheets, but for both A levels, that was background reading, which would be excellent examples, and they are held together by hydrogen bonds. Then for the tertiary structure, you would be mentioning, right, well, if it's it tells you it's a globular protein, so the tertiary in the question tells you it's a globular protein, so the tertiary structure is the further folding of the polypeptide chain, and there are 3 main bonds that hold it together. That is disulfide, ionic, hydrogen bonds, and then there's also the idea of hydrophobic and hydrophilic interactions. And it says here about hydrophilic R groups tend to be on the outside of the hemoglobin molecule, whereas the hydrophobic R groups tend to be on the inside of the hemoglobin molecule. Now, if you think about it, all of those 3 up until this point pretty much would give you the answer for any question about a protein. So any globular protein. So if you're asked about insulin or any hormone, that would be how you'd answer it. If you're asked about collagen or one of the fibrous proteins, it would be very similar answer, apart from the fact that you'd say that it's got very little folding and coiling in the tertiary structure. And then if we finish off, we go on to the quaternary structure, the 4th level, which would be specific to hemoglobin, and a lot of the fibrous proteins, if you're asked about that, don't have a quaternary structure because they just have tertiary. But quaternary structure for hemoglobin is there are 4 polypeptides, 2 alpha and 2 beta chains, as I mentioned a few moments ago. There is each one of those polypeptides has 1 heme group, or there's 4 heme groups per hemoglobin molecule, and that has a prosthetic group with an ion, okay, or you could call it Fe2+, which accepts the oxygen. So I hope that's been useful. Here's some more questions that you should be able to answer if you understand what we've gone through today. So thank you very much, and I'll see you next time.